The overall goal of this experimental procedure is to investigate the Coronin A-dependent early starvation response of Dictyostelium discoideum.
This method can help answer key questions in the field of developmental biology, such as the identification of signaling pathways and factors involved in the early development of Dictyostelium discoideum. The main advantage of this technique is that it has the potential to provide a tool for automated and high-throughput screening of development-inducing factors and proteins involved in early developmental signaling.
Generally, individuals new to this method will struggle because of multiple variables inherent to the assay, such as cell densities, time of adherence, and time of development.
To begin this procedure, grow DH1.10 cells or Core deficient cells in an Erlenmeyer flask containing HL5 medium at 22 degrees Celsius in a shaking incubator at 160 rpm. Keep the cells at a density between 1 × 10⁴ and 2 × 10⁶ cells per milliliter. To examine cell aggregation, harvest 10 to 50 milliliters of log phase growing DH1.10 cells or Core deficient cells generated in the DH1.10 background. Centrifuge them for 3 minutes at 400 × G. Then wash the cells twice in BSS. Afterward, count the cells using a hemocytometer. Subsequently, plate the cells in a 24-well plate. Allow them to adhere for 1 hour at 22 degrees Celsius in BSS.